All right, coronavirus replication, part 3. Let's go. Okay, so now for the nitty gritty of it all. Let's see. So, first, let's talk about the NSP9 cycle. And so, NSP9 is the ninth part that's going to be processed out from polyprotein 1A, or 1AB, honestly. And this one has a special role because it's kind of going to start things off in two different ways. So it is the protein that's involved in initiating both of the major activities, which are making RNA and then capping the RNA, which is a big deal and a lot more work than you would think it would be, for sure. Yeah. So first of all, let's meet our participants here. So you've got NSP7, and that's the one that I told you. It doesn't do much. It sits there, and it's a nice pink color in this, whatever that is. I don't know what that is. Washed out. Yeah, I don't know. You've got NSP8, and there are two copies. That's what the A and B are. They're two copies of the same thing. It's just that they adopt a different shape. One of them bends a little bit. NSP8 looks like a golf club. That would be the head. This would be the shaft. One of them is a little bit bent. The other one is pretty much straight. And yeah, that's fine. And so the bent one sits at a particular part on the complex, and the straight one sits next to it on the complex. Fine. Then we've got these two parts, this sort of sickly green-yellow puke color part here, and we've got the nice orangey part. Oh, that's a good color. Look at that. These are both parts of NSP12. So NSP12, NSP being non-structural protein, contains both the RNA-dependent RNA polymerase, or we can just call it the polymerase. It's the main virus polymerase. And it contains another domain, which is to say another part of the same protein that sort of folds up on its own and does its own job. But they're attached. They never get separated from each other. So it's like, I don't know, like you have a left hand and a right hand, and they're both attached to your body, and they can be doing different things, dribbling a basketball and playing piano or whatever. It's fine. This one is called the NERAN, and NERAN stands for something that we don't need to remember. But this is a special enzyme that can add one nucleotide onto NSP9. So NERAN puts nucleotides onto, it loads up NSP9, and then NSP9, depending on what nucleotide is loaded, will go out and do different things, which is kind of cool. So NERAN and NSP9 are a team. Oh, best friends forever. And the polymerase is then going to use one part of our little team. All right, so first step is that NSP9 is going to bind to the NERAN, and this D218, that's telling you that there's an aspartic acid at position 218. I don't remember that without looking at this drawing. I do not expect you to remember that. Just know that there's a NERAN domain, NIRAN. So it's going to bind here, and it is going to take, so where it says NTP, that means it's like ATP, CTP, GTP, or UTP. It's one of the nucleotides, and since we're doing RNA, we have a U instead of a T there. Yeah. And so NERAN can put any nucleotide on, at least in theory. It seems to prefer putting on U nucleotides. So that seems to be its most common thing to do. But there is a real possibility that it can put on a different nucleotide, and that's okay. Yeah, that is just fine. So we just say it puts an NTP on because it puts one of those on, and we're not specifying at this point. The guy who wrote this review is being very careful, and honestly, he's careful and he's usually right. So yeah, maybe we should all be careful like this. When we put it on, we're going to lose two phosphates. So that's the little PPI. It's pyrophosphate inorganic, pyro, PYRO. Phosphate. It just means two phosphates stuck together. and they're not attached to any carbon, so they're inorganic, even though they came off the ATP or whichever nucleotide it was, UTP. And so now there's going to be one phosphate left, and that's how we're going to attach to the NSP9. And it's actually attached to the N-terminus, the amino terminus, which is the very first amino acid in NSP9. And so that's where we stick a nucleotide. So NSP9 is a little carrier. It's like a little truck that you can load with one nucleotide, put one little nucleotide in there, and then it's going to go off and it's going to do something. So the first thing it can do is it can take its little one nucleotide over to the polymerase, spot 1 over here. And when it gets there, it's going to prime RNA synthesis. So you remember how polymerases need a little help. They need a primer that they can extend, but polymerases can't make a primer. They just don't know how to do that. They're bad at it. Yeah. And so this protein, NSP9, acts as if it was a primer. And you would say, now, wait a minute. I know primers have to be either RNA or DNA, and they base pair. Not this time. This is a protein that's going to base pair, we think, with a specific bit of RNA. And it's got a nucleotide on the end that you can build onto. And yeah, it's a primer. That's it. Yeah. It is a protein primer for RNA synthesis. Yeah, weird concept, but you find this in several viruses. And so coronaviruses turn out to be one of the viruses that works this way. Okay. After that, then the NTP on the end will stay there, and NSP9 will be attached to it, and we'll just keep adding nucleotides on, and we'll copy the entire genome, positive or negative strand, whatever, but it's going to start with an NSP9. So NSP9 starts it. It starts RNA synthesis. And the polymerase, the big puke green thing right here, that's actually going to do the activity. Now, we think it's also assisted by NSP8. So we think NSP8 may help to extend that one nucleotide of U to make it several nucleotides long. How many? Nobody knows. If you just take NSP8 and you give it some nucleotides, it'll copy 30 or 40 or so before it just runs out of steam. So it works like a polymerase, but it's not a very good one. And so we think it's probably just there to barely extend that U and get the primer a little bit longer so that it can be passed over to the main polymerase in NSP12. Okay, after that, NSP9 is going to get cut off from the RNA, and it gets recycled. And so once NSP is recycled, it's going to, before it gets recycled, it's going to deliver this RNA back to the NERAN. Because remember, NERAN and NSP9, they're a team. And so there's another function there. There's a little pocket inside where it can hold an extra nucleotide, and that nucleotide is going to get flipped around backwards by NERAN and put on, and that's going to be the cap. That's hard to conceptualize. We have a couple slides with pictures coming up. So for now, we're going to say NSP9 gets a nucleotide on it, probably a U, and that serves as the first primer for RNA synthesis. It's probably extended by NSP8, and we're not sure which of these two copies, but at least one of them, maybe both. And then it's passed over to the polymerase, and then it will be extended to copy the entire genome. So it just makes a little primer a little bit longer. Then the NSP9 plus the genome is going to go back to NERAN, and probably it never left. Probably it just hangs out in this vicinity. And now it's going to swap out, and we're going to change out. We're going to take off NSP9, and we're going to put on a backwards G nucleotide to be the cap. Yeah. And that's the capping process. And now NSP9 is free, and it doesn't have anything attached to it. And so where's it go? It's going to hang out on NERAN, which it kind of already is a NERAN. And it's going to get loaded up with another NTP on its tail. And it's going to get sent out, and it's going to get elongated and turned into a strand, and then chopped off and replaced with a cap. And that's the NSP9 cycle. It's the circle of life right there. Yeah, cue the music. Oh. Yeah. All right. So that's how we do the startup process, how we start RNA synthesis. After that, the polymerase carries out RNA synthesis. It just goes. Once you've got a primer started, once it's got something to copy, there's none of this partial strand synthesis with strand transfer, jumping around stuff. It starts at one end and it stops at the other end. It just copies. Yeah. NSP12 is what we call processive, Processive, in that it just goes through and it just reads and copies what it sees. Yeah. All right. So this is the NSP12 polymerase with all the other things that sort of make it work. So the best friends of our polymerase, our favorite polymerase, are NSP7. Now you can see one tucked in there, and there it is poking out the top of that. We've got NSP8, so this is the golf club side of one of them. That's the bent one, and then there's another NSP8 hiding around the back there. And you can see its club handle poking out over here. And those are going to form, it's kind of like a tunnel where the newly synthesized RNA is going to come out. The RNA that's coming in is going to have to pass over this guy. And so this is NSP13, and this is the helicase. And so a helicase, you know, is going to unwind double-stranded nucleotides. So in this case, if the helicase is coming along and it sees any double-stranded nucleotides, there we are, a nice little double helix, twisted up, it's going to peel one of those off, and it's going to keep the minus strand, so it can make a copy of that, basically. So yeah, it peels off the plus strand and sets it free so it can go out the pore and go lead its best life. And it's going to keep the minus strand nice and close and protected. And it's going to feed that minus strand into the polymerase. It's like feeding it into the mechanism. And then once it's in the mechanism, the polymerase does its usual, fingers, palm, thumb thing, and it opens and shuts and puts little nucleotides in, just like that. Just like that. Yeah. Now, NSP13 has a couple of domains in it, which are just functional parts of this thing. So it has two of these modules that are called Rec-A. And if you work with bacteria, you'll run into Rec-A. It's known as a recombinase. In this case, it's just a particular fold, like a particular shape of protein that was probably stolen from bacteria millions of years ago, because most everything is. And so there are just two copies of that. There are two Rec-As that make up the helicase. Which is fine. And there's also a zinc binding domain. That's the atom zinc, Z-I-N-C, binding domain. So that's the ZBD. All right, the second Rec-A domain, Rec-A number 2, sticks up. And they say it sticks up like a thumb. Now this is different from the fingers, thumb, palm of the polymerase. So this thing has two thumbs, and I wish they had called it something else, but it kind of looks like a hand doing that if you see up here in the corner. So that's why they call it the thumbs. Yeah, what's this? This thumb, or Rec-A number 2, is going to be the thing that's actually going to it's basically pulling the RNA along. So it's going through and making sure that we're splitting off any plus strands and throwing them away so they can go off and be translated or be packaged. Feeding the minus strand down. And also, if there's a mistake, so if our polymerase puts in the wrong nucleotide, then this is the part that's going to say, whoa. The thumb is going to come in and say, whoa. We're going to put a thumb on it. It's going to slide the RNA back, just pull it back, using ATP to actually pull the RNA back. And then it's going to give the polymerase a second chance. It's going to get a chance to chop out the bad nucleotides, put in the right nucleotides. And so in this way, the virus will be able to keep its mutation frequency down. So this is part of the error-correcting machinery and process. The other part of that is going to be an enzyme called NSP14, which has an exonuclease, which is a proofreading enzyme. Yeah, that takes out a misincorporated nucleotide, puts it in. So there you go. Those are the parts of the polymerase and helicase. All right. Now we'll save this for the next part because that's good. We've got the polymerase down. I'm happy with that. All right, part 4 coming up. See you there.